So I want to, in this presentation, really start a little bit at the beginning about why gene therapy, tell you a little bit about the progress of why somebody like me is just so excited about gene therapy. And then really talk about what I'm really here to talk about, which is kind of, I also, it's like next level gene therapy, next gen gene therapy using genome editing with lipid nanoparticles. So I do want to start at the beginning though. Why gene therapy? Because there are thousands of genetic diseases out there. There's about 1 in 40 people in Canada that are affected with genetic diseases. And they account for about 40% of all infant deaths in places like Canada and the US. And the problem is there's nothing to do for these patients. We can sequence their genomes now, we can tell them exactly what's wrong, but there's nothing you can do. And for those few cases where you can do something, the cost can be millions of dollars a year. Many of the new enzyme replacement therapies are $300,000, $600,000, $700,000 a year. And so for me, what encourages me about this is that there are millions of Canadians that are affected, but for me, what drives me in my research is really the individuals. And so all of these people have genetic diseases that are progressive that will eventually kill them. Some sooner than others, like Michael and Makeda probably won't live till they're 20 if they're not treated in some way. And what they all have in common is they all have an underlying genetic disease-causing mutation. And if you can fix those, then you can potentially treat all these diseases. And that's really the underlying rationale for gene therapy. You could treat these diseases right at the DNA level. And it's really not a new idea. It's been around 75 years was the first use of the word gene therapy. And so for me, my start got started at McMaster, and really just 2 lessons came out of my entire PhD. One was that in vivo delivery for gene therapy is key. We can do all we want in vitro, but on the delivery side, so this is not, I'm not saying anything negative about Sabrina and her amazing results, because she's doing different questions. But on the delivery side, in vivo, using a model or a mouse or a human is key. And what I also took from this research is just that the impact of gene therapy can truly be remarkable. And this is from my PhD, and on the right is, on the left is the mice before they were treated. They had this terrible lysosomal storage disease, and I would treat them with this very crude gene therapy, and they would you could just tell by looking at them that they were different. And when you actually just measured how much they ran, it was really striking. So I was hooked, and I made it to UBC and BC Children's Hospital and started a project on gene therapy for LPL deficiency. So LPL, you don't need to know much about it. It is so complex, the regulation of LPL. In fact, there's a researcher here, Brian Rodriguez. His whole career is studying the complex regulation of LPL. It basically controls where your fats go in your blood, to your heart, to your liver, to your adipose, to your skeletal muscle. But for us, this disease is caused by deficiency of LPL. So these patients develop really high triglycerides in their blood, so much that it will just try to come out of your skin, or it will go into your pancreas, and it can cause this fatal pancreatitis. And it's truly a Canadian disease. There's no other country in the world that has a higher incidence of this disease. And you can blame this on the Scottish and French settlers from the hundreds of years ago. And so the progress towards a treatment for this, I love this picture, because this is them celebrating the discovery of the gene for LPL, and they found the first mutations that linked this disease with LPL deficiency. So they linked type 1 hyperlipidemia. It's really just a silly picture, but some of the people are still involved in this. And for example, John Castelline is at the far end, and Michael Hayden. So 35 years ago, so what have they achieved in that time? So they discovered that lipid-lowering drugs didn't work, enzyme replacement didn't work. The only way to treat it was to basically cut out all dietary fat, and that's not actually so easy. So they, by discovering the disease-causing gene, they thought, well, we could treat it that way. And so they tried a lot of different approaches for this too. So none of them worked. They were all ineffective or toxic or short-term duration. And so I was in the right place at the right time, and I was really excited to work anywhere with this new type of viral vector coming out called an AAV. And it's the smallest known virus that infects human cells. People have spent their entire careers developing this into a vector, so I was just at the right place at the right time, and we learned how to use this AAV. So I'm not here to talk about that, but I just wanted to fast forward about one key lesson I learned in that, and was that when I started, we had to choose what type of serotype. There weren't that many. There's now hundreds of different serotypes of AAV. But of course, the in vitro results were really clear. We should use AAV serotype 2 because it had such high expression or high transduction of cells in vitro. But I had learned from my PhD that it's the in vivo delivery that is so key, and we found that the AAV2 was basically useless in vivo. Whereas AAV1, which was the worst one in vitro, worked by far the best. Like, it would just transduce so much of the muscle. That's what we were trying to target. The entire leg would go green and literally just glowing, just in a living mouse. And so in this study, we showed that we could completely reverse the hyperlipidemia of this horrible condition. We did it in a large animal model, and then we managed to get into a clinical trial. And I put this slide in because we thought we were pretty smart using these fancy roller bottles at the time, but it cost $1 million to make enough of this for one patient. The results of that trial were that no adverse events occurred, and it seemed to be relatively efficacious. But we realized we probably needed to do a higher dose. And so they did a phase 2/3 study at up to 10 times higher doses. And again, it worked okay. It reduced the triglycerides out of the dangerous zone for patients. And a 6-year follow-up study showed that it prevented all serious pancreatitis and reduced the use of healthcare resources significantly. And so this company that my supervisor had helped then create, I was never part of this company, but they eventually became bankrupt trying to get this drug approved. They tried to get it approved like 3 times. And then finally another company bought it up, and it finally made it and got approved because it's a little bit scary using a virus to try to treat a disease. But it was very exciting to be a part of that project. The patients, though, this is what really impacts me, is the patients talking about how impactful this was for them. I won't try to state their statements, but it was a really striking effect. And there's many patients still in Canada that benefited from this. But in 2017, this very expensive drug got removed from the market because nobody could pay for it. And it was interesting. It actually was on the news, which was good, but also sad. And the problem was there was really the companies or countries like Canada and Germany and the UK, they didn't have mechanisms in place, especially at that time, to pay for such expensive single injection drugs. And I just put this slide in because the work that I'll tell you about for the lipid nanoparticles, I think, can be much, much less expensive. But the problem is that Canada spends right now about $33 billion on all drug spending. Since then, since 2012, there's been about 45 new gene therapy approved since Glybera, and there's hundreds more. And as Peter mentioned, there's 300 LNP only type nanomedicine gene therapies in the pipeline. And so this is an extremely conservative estimate of the cost. It's going to be $30 billion a year just for these gene therapies. But this was really a very small study looking at just 5 indications. So it's really not. It was the highest number I could find, but it's not a really realistic number. So we can double the healthcare spending to treat those 5 more indications, or we can try to lower the cost of the drugs or hold lotteries for access to these medicines. And in fact, it's the lottery option that seems to be coming up in the UK for access to Zolgensma, which is a $2 million treatment, that's not what we're looking for. And so I really like this quote from Francis Collins saying that we need a more transformative approach. And that's really what I think this nanomedicine can do. I'm going to just really briefly tell you about the 2 approaches, though, because it's a little bit conflicted, because I do love AAV, but I also love the LNPs with Peter and others. But So one approach is just, could we actually just redevelop that AAV and maybe make production facilities that would make it low cost for other Canadian researchers to use? And so this has been a really fun project to be part of. It's been about 5 years of work trying to make a cheaper AAV. Neil is a postdoc that works with me that basically does everything for this. He's done such a better job than I ever did as a postdoc. So, and then Danica is our NRC sort of contact. And just to be very brief about this is that Neil, in 5 years and one slide, has made it like a 50-fold improved version of this AAV, which is wonderful, and we just published it. But let me tell you about the other approach. So this is really next level gene therapy. This is talking about gene editing and using lipid nanoparticles for that. So why would somebody but like me? I mean, AAV works great. Why am I so interested in genome editing? So the pros, the same as AAV, it's permanent. But what it can do, this is the big differentiator, is that it can restore the precise regulation of the gene that you're trying to correct, because you're restoring that precise original mutation, turning it back to wild type. And that's so important for genes like LPL. And then on the cons side, there is a big risk. I mean, the main con is that this is really difficult to do. It's really challenging. And if you don't do it perfectly, you're going to get these off-target edits in the genome. So my main message there is that it's just not easy. It's not going to be easy. And as a reminder, I love to show this slide in teaching because we all forget how big our genome is. And imagine if you're typing it out in 10-point font, it literally stretches all across Canada. And a single nucleotide difference is the difference between developing cystic fibrosis or some other life-threatening disease or not. And in order for gene editing to actually work, it has to somehow scan that entire genome, find the exact mutation and fix it and not mess with anything else. And we have a lot to be thankful for in the gene therapy field from Manuel Charpentier and Jennifer Doudna for their discovery and their use of CRISPR as a tool. And so this is the more traditional approach of CRISPR, where all you need is the two things, the Cas protein and the guide RNA. They come together and it scans the genome for an exact site that guide RNA sequence aligns with. And it's that programmable part, that green part on this slide, that's the beauty of CRISPR. And then it causes a double-stranded break, and people can use that in different ways. But I'm not going to be talking about that approach. I'm going to talk about new versions of CRISPR, or basically new versions of genome editors that are based on CRISPR. And these were all developed by David Liu at MIT. I'm going to really mostly just talk about base editors. And Peter sort of introduced base editors a little bit at the end of his talk about that Verve study, and I'll finish with that as well. But another type is prime editors, which I won't talk about. But these base editors, I'll just go over how they work very briefly. So they can precisely repair they can do that task, which I just find so amazing. They can find that single sequence that aligns with the target on the guide RNA and edit it. And so it does it because they've added these DNA modifying enzymes onto the Cas. And so again, you need your guide RNA. It forms a complex with this base editor protein, and it scans your genome, finds the exact site, and now it's not cutting the DNA, it's just modifying a single nucleotide. That's the ideal outcome. So a single nucleotide is turned back to whatever you want it to be. And so getting to what we're trying to focus on here at UBC and PharmSci is, can we improve the delivery and effectiveness and safety of these new genome editors, as a tool for genome editing. And that's a lot of things to focus on. Liam has a poster here on the effectiveness side. Tessa has a really great poster on a really nice model system she's made that's looking at the safety side. And I'm going to just talk about the work from Alex and Tyler and Tiffany and Alice about using lipid nanoparticles as our delivery and trying to make them better. And I really, again, cross out AAV, because even though I love AAV, the use of AAV in genome editing is not at all well suited, because AAV just goes in there and stays there forever. So of course, in a model system, it would work well. It would be great. But as a therapeutic tool, it's not an appropriate vector. We want something that can efficiently deliver our genome editor to wherever we want it to go, and then just do that edit and then be gone. And I reminded myself that in vitro doesn't equal in vivo, so if we're trying to focus on the delivery side, we got to keep that in mind. And this also applies not just to things like AAV, but also to lipid nanoparticles. And this is a paper from James Dahlman, who in many, many figures basically said that in vitro LNP delivery does not predict in vivo delivery. So you have to have either like a really good model, like a really good cell-based model system, or you have to do this in a living animal. And so we developed these mice, which our goal was to develop a model that we could rapidly evaluate really precision genome editing down to a single nucleotide level. And so we made these luciferase mice that have the luciferase gene in every cell, but they have a particular mutation that knocks out the signal. And so if we can repair that one mutation, it should turn on the luciferase. And you should be able to then measure that. And you can use this then model to evaluate changes to lipid nanoparticles or other delivery systems. You could use it to test out new genome editor formulations. There's a lot we can do with it. And so one nice thing too is that you don't have to kill the animal when you want to take a measurement. You can just keep measuring it from these really sensitive imagers called IVUS imagers. And so here's us getting ready. We've made the mouse, and we want to see if we can fix this mutation. And so we turned to Peter and his colleagues, who were, just without their technology, we wouldn't be able to do any of this. And so we've incorporated a lot of the things that they've spent their careers developing. So again, sort of standing on the shoulders of giants to make this possible. And so in this case, we used some of the LNPs that were just very simple, sort of off the shelf, the ALC 0315, the Pfizer type formulation of LNPs. And in this case, we injected them intravenously, and they all go to the liver, and that liver, they precisely edit the mutation in the gene, and they turn on the luciferase signal, and the livers light up, and you can see it. It's just a beautiful result. And so after 4 weeks, I was really excited. It was still working. As Peter said, it should be simple. Like gene therapy, the idea is simple, and we want like boring results that just show the same thing every week. And then this is exactly what we're seeing. So this went out to 4 months. We injected this into the leg muscle of mice. So, 4 weeks. And then I'll just give it 1 second. And then again, it's kind of boring results, but beautiful results. It's just so amazing to see this. So this goes out to week 30, and then Alex sent me another update. So now it goes out to week 72. But again, there's the same result over and over and over, because this is, you've permanently edited that single mutation in the luciferase gene. So what are we trying to do to make this better? Because we aren't actually that great at it yet. So we've been trying to use new formulations to improve the efficacy and the safety. And so this really builds on Dr. Miffy Cheng's talk this morning, using her new formulation of lipid nanoparticles. And so these are figures that were in her paper that she showed this morning. And just in summary, it was this new formulation shows about a 10-fold improvement. So could we use these to deliver genome editors? And again, just using the same mRNA base editor and the guide RNA, put them in mice and use a low dose, and you can see there's a striking increase in the luminescence in the livers of these mice. It's about 10-fold. And then if you actually measure the luciferase in the livers, or you measure the DNA, you see a 10-fold improvement in the gene editing there. So another approach is, can we try to get these LNPs to deliver genome editors outside of the liver? Because the liver is beautiful, and there's literally thousands of diseases that we could probably treat today, and we will end with one, but by treating the liver. But there's so many other diseases that we also want to get into different tissues, and I'm really want to get things into the CNS, into the muscle. And so we're trying these new formulations with Peter and team, and this example is from long circulating lipid nanoparticles, and you can see the effect of of these. It really does spread it throughout other tissues, and yeah, it's just very preliminary results, but it's definitely looking very good. Another approach is, what if we can combine these ideas together using these long circulating LNPs with other just really quite basic optimization of genome editing, because we'd never tried to optimize some of the components that are stuck inside these lipid nanoparticles. So we tried different ratios of the guide RNA, and so these are using these long circulating LNPs at different guide RNA ratios, and you can see it's such a striking effect of just this simple change, but again, using these really state-of-the-art lipid nanoparticles from Peter. It's, yeah, it's a really nice result. And so that's my talk. The summary is that I'm so excited about the use of genome editing to treat literally thousands of different diseases and different disease-causing mutations. And what is so, it's such a powerful and efficient approach, because you can use the exact same mode of delivery if you wanted. You could use almost the same cargo. The only difference is a small 20 nucleotide difference in the targeting. And so our sort of dream is, and I think we're getting there, is, could we deliver this genome editing into all of the cells of the mouse and eventually into a human? But of course, it would be great to go to other tissues as well. We're trying to do that as well, so trying different approaches. And I just want to end on this slide, because I think it's really important to remember what we're doing this for. All these patients with these progressive, fatal genetic diseases, and there's nothing that we can do for them now. And Peter stole my thunder this morning with these amazing results that were just published 2 days ago, were presented earlier this month. But it's so exciting to see that people are actually now beginning to use these genome editors. This is a base editor, as Peter mentioned, to treat familial hypercholesterolemia, and it's really, really amazing results. So I'll end it there, but just to acknowledge the really wonderful team. A lot of them have posters here today that work with me here at UBC. So thanks very much.
Okay, well, thanks, Colin, for the great talk. Are there any questions? Peter has a question.
Oh, hi, Peter. I can only predict, and as you know, I liked your quote this morning about predictions, but I would personally be happy to get this into all tissues, into every single cell in the body. That would be the easiest way. Then you could use the same delivery system for every single disease out there. And I'm not worried at all about fixing the mutation in a tissue that doesn't, isn't really affected by the disease. But I guess the concern would be you'd have to make sure that it was a safe enough system. And of course, that's the same, even if you're just fixing just the liver or the muscle, it has to be safe. So.
Are there any other questions? Maybe a question from us. Diana?
Hi, Diana. Yeah, no, I really want to work on that with the genome editing as well. Actually, we've been funded to do that, so we're really excited about it. So, because I mean, it's probably I'm hoping the gene editing. Well, I always would say I'm hoping this isn't. We're not going to see this for 10 years. I'm different from Peter. I'm a little bit more cautious. I think Peter's ready to start a company the next day, but I'm quite yeah. It certainly has. But this stuff is amazing to see that there's clinical trials now, and I kind of glossed over it, but in this clinical trial, a patient died out of a small number. But it's because the disease they're treating is so severe. So they carefully evaluate every serious adverse event, and they say this was just because of the patient's serious underlying disease. So all of these patients are going to die if they're not treated. So, yeah, Peter. I've done it. I've done a non-detailed cost analysis. Yeah, I've done the non-detailed one where but I think it would be like 10,000 per person. So that's like nothing compared to AAV. I mean, even AAV today is pretty expensive. It's all about scale, though, right? It's hard to judge. But I really just want to see like a guide RNA synthesizer in the basement of the hospital, and then everything else, you could just have it in the freezer. It'd be set.
Is there a situation where AAV might be needed and the lipid CRISPR could not be successful, or is it the lipid RNA has been presented as a universal solution, or with CRISPR, but is there a place where it conceptually can
like
fail?
AAV versus?
Yeah.
AAV is a wonderful approach. There's hundreds of trials underway that are working towards clinical approval, and they're treating really devastating diseases. So I'm not being negative to them at all. I think they're trying to fill a need, but I think gene editing overcomes a huge issue, which is this restoring the regulation of a gene. I mean, it's an absolute dream, and it's so important for many, many conditions.
Maybe something involving like the blood-brain barrier, or situations where there's just advantage in molecular versus the particle delivery.
maybe.
I don't know. I'm just curious. I had another question, which is the non-correlation plot you showed.
Yes, I
feel
was
obliged to
try
respond.
to introduce it.
I'm
gently,
curious
'cause-
if the way to unite that with some of the other work that has shown correlation more recently is maybe that it's important to take formulations that already pass certain tests, like motivated from successful delivery, and then work on like next stage optimization in this kind of funnel. And there, maybe that's where the correlation is good, whereas the sort of blind search might, at a macroscopic scale. Is that a unifying perspective for the different papers on this? Or?
I don't know. I can't really predict. I'm not sure.
Okay. Well, maybe that's a dinner conversation.
Yeah.
But